What are intrinsically disordered proteins? Proteins are like tiny machines in our bodies that help carry out almost everything cells need to do, like building structures, communicating, or cleaning up waste. For a long time, scientists believed that all proteins had to form into a specific stable 3D shape to perform their functions, like tools with specific shapes that match their jobs. For example, a protein might look like a key that fits perfectly into a lock to perform a specific task. This idea, known as the structure function paradigm, dominated biology for most of the 20th century. Proteins without a clear structure were thought to be defective or useless. But in the 1990s, this idea began to change. Scientists discovered that some proteins, or parts of proteins, don't fold into fixed shapes. Instead, they stay flexible and disordered, like a piece of string or spaghetti. The term intrinsically disordered proteins, or IDPs, was introduced because these proteins lack a stable 3D structure. And it turns out that these proteins weren't broken at all. They were doing important jobs. So, why are they important and how do they work? Instead of having a fixed structure, IDPs are flexible and shapeshift, like spaghetti or a ribbon floating in water. IDPs work by wrapping around or sticking to other molecules in many different ways. Imagine a piece of Velcro that can mold itself to any surface. This adaptability allows IDPs to interact with many different partners, making them excellent at multitasking. For example, IDPs are essential for things like cellular processes, including cell signaling, regulation, gene expression and transcription, cell cycle control, guiding cargo and membrane interactions. They also have the ability to form membraneless organelles and scaffolds for assembly of other proteins through organizing complexes and phase separation and compartmentalization. They're also crucial for immune system function by antigen recognition and cytokine activity. They have a role in stress response pathways as well as an evolutionary advantage by adapting to cell stress and being drivers of evolution. So, what happens when IDPs go wrong? While IDPs are essential, their flexibility can sometimes backfire. If they misbehave or interact with the wrong molecules, it can lead to diseases like prion diseases. Some IDPs are linked to prions, which are proteins that can fold the wrong way and then cause other proteins to misfold also. This chain reaction can lead to serious diseases like mad cow disease or Creutzfeldt-Jacobs disease. And they're kind of a scary example of how the flexibility of IDPs can sometimes go wrong. Alzheimer's disease is another example. Misfolded proteins, some of which are IDPs, can form these kind of clumps in the brain that interfere with normal function and cause memory loss. Another big example is cancer. Cancer happens when cells grow and divide uncontrollably, leading to the formation of tumors, and IDPs can play a big role in this process by switching genes, or like oncogenes on and tumor suppressors off, or by triggering the wrong signaling pathway at the wrong time, which can cause cells to ignore the normal rules for when they're supposed to divide or stop growing. IDPs in the dark proteome remind us that biology is full of mysteries. They show us that even the messy parts of life can have incredible purpose, and not everything in biology needs to be neatly organized to be important. These shape-shifting proteins are like creative problem solvers, showing how flexibility can be just as valuable as precision. Studying IDPs could lead to breakthroughs in medicine, especially for diseases like Alzheimer's and cancer. So even though they don't play by the usual rules, IDPs are crucial for life and show us that sometimes being disordered can be a good thing.